Good morning. This is the part two for your lesson today. So we're going to be doing codominance and multiple alleles. Let's get started. So let's make some observations to jump in for this topic. So what patterns are you noticing in these individuals? Okay, if you need to pause and look at the video for or the pictures for a couple of seconds, please do. But what patterns are you noticing? What do these all share in common? What's kind of unique? So I hope what you're noticing is that when you look at these individuals, they're expressing two things at the same time, like with our cow, brown and white, with the flower, pink and white. With our little hen, there's a white hen, but then there's one that's black and white. Or like this is one of those little bluey dogs in the bottom corner. It kind of has like that like white and kind of like grayish color and it kind of looks blue. Or a calico cat, okay, is expressing some black and white and orange. So some really interesting things that we're seeing here. We're organisms expressing two things at the same time. And that's what we call codominance. So in your packet, make sure you write down your definition. With codominance, two alleles can be expressed independent of each other. We looked at 4 o'clock flowers when I was introducing this idea of non-Mendelian genetics, a pink with a white. And in this case, all of the offspring were pink and white. Okay, so if you look at my Punnett square, one of the unique things when we're doing this is we tend to use superscripts to differentiate the alleles. We want to treat them kind of like they're all dominant. In this case, it's codominant, so they are dominant alleles. So we have a capital C to denote that it is dominant, and then because there's two dominant alleles, I'm going to differentiate that one, and that would be the pink allele, and that one would be the white allele. So use those superscripts. So if you look at that, the offspring, okay, they have those two dominant alleles expressing both pink and white at the same time. So let's try a couple of practice problems. But the thing that you kind of tend to get with codominance is you tend to see multiple alleles. So we're going to do a couple practices with multiple alleles. In this case, we're going to look at some rabbits. This one just happens to have one dominant allele, but it has many. Oh, where'd my little albino thing go? It should be a CA here. But multiple alleles means that there's a single gene with many possible alleles that can be inherited in many patterns. So we're not just dealing with two alleles for one gene. Now we have many alleles. And so in this case, we're going to look at some rabbits. We have agouti, we have chinchilla, Himalayan, and albino. And agouti happens to be more dominant than chinchilla, and chinchilla is more dominant than Himalayan, and Himalayan is more dominant than albino, which is my little friend down here. So whenever we're doing codominance and multiple alleles, we tend to see that there are many different alleles and many different patterns. So we'll come back and do a little bit of work with both of these, but I do want to play with multiple alleles for a couple minutes and practice with our little rabbits. So in your packet on page 23, we're going to do a couple practice problems. In cross one, we're going to cross a purebred agouti rabbit with a purebred albino rabbit. Okay, all the F1 offspring are agouti, so see if you can map that out in your packet. For cross two, we're going to cross a purebred Himalayan rabbit with a purebred albino rabbit, and all the F1 offspring are Himalayan, so see if you can map that one out. And then we're going to cross the offspring of cross 1 and 2. And they have 12 kits, which are the name for baby rabbits. And we're going to think about how many we would see of each phenotype. So let's play around with like this idea of multiple alleles, and then we'll jump into some examples that have both multiple alleles and codominance in a second. So what you should do now is pause your video and see if you can finish number 5 through number 14 in your packet. Okay, so you should have done 5 through 14 in your packet. I'm just going to walk you through some of the highlights and make sure you feel good about how to solve these problems. So when we're thinking about our rabbits, for our first cross, we're crossing a purebred agouti rabbit. Purebred means homozygous, so agouti would be, agouti would be big C, big C. and we're going to cross with an albino rabbit. So this is little ca, little ca. And so if we set up this cross, each parent can give one allele, not both. So if I set up this cross and make myself a little Punnett square, this parent, the agouti can give big C or big C. The albino can give little ca, not both. And so if I bring these together, okay, I'm going to see that all of the offspring are agouti, and that's true because they have that big dominant big C. So what's the dominant allele? Would be agouti. What's the genotype of the kits? So there's a siren happening. So the genotype of the kits would be big C, little c, superscript A. And what's the phenotype? Okay, they are all going to be agouti. Let's do cross 2. Okay, so cross 2, we're crossing a Himalayan rabbit, and what's interesting about Himalayan rabbits is they have color, but the color is only restricted to their ears, their feet, and their nose. So otherwise they are completely white. So a Himalayan rabbit, and so if we think about our Himalayan right down here, little c, superscript h, ch. Okay, ch is going to be the genotype of my Himalayan rabbit. My albino rabbit, C superscript little c superscript A. If I cross these little friends together, okay, my Himalayan rabbit can only give this allele. My albino rabbit can only give this allele. And if I bring them together, okay, I have two alleles here. I have a Himalayan allele and an albino allele. But if I look at kind of my in this case, I have kind of a spectrum of dominance. If I look at my Himalayan allele is more dominant than my albino in this case. So all of these friends would be, the Himalayan would be more dominant. The genotype of my kits would be little c superscript h, little c superscript a, and what would these express? Okay, they would all express Himalayan. If I cross the offspring of these two crosses together, that's where it gets a little bit interesting. So if I cross the offspring, so now I'm crossing big C, little ca by chca. If I set up my Punnett square, I'm going to do it over here. I must have enough room. So big C, little ca, little ch, little ca, and I bring these together. This offspring that was just created would be agouti. CHCA, this one would be Himalayan. Big C, little ca, this one would be agouti. And CACA, this one would be albino. So if I have 12 kits, okay, I would expect to have half of them be agouti because they have that dominant allele. So I have 6 agouti, and I would have 3 Himalayan, and then I would have 3 that are albino. And so that's how you can work with multiple alleles. You really just kind of have to think, what's more dominant than the other? The next one that we're going to head into, just to do some, to wrap up our practice and kind of link together this idea of multiple alleles and codominance, is blood type in humans. Blood type is super fascinating, and it kind of puts all of these things together in a really cool way. So let's go to blood type. And if you go to your packet for number 15, I have a little description of blood type, and you can kind of think about what this might look like. So what's cool about blood type is we're thinking about red blood cells here. And red blood cells have surface antigens, and that basically is just proteins that are embedded in the membrane of the cell. And this is coded for by a single gene for this, this specific type of, like the ABO blood type. So cells can have A and B surface antigens, but it's possible for codominance to exist where they can have A and B antigens at the same time. and type O would be no surface antigen. So we're going to represent these with these different alleles. The A allele is big I superscript A, big I superscript B. Note that I have two capital letters. These two are going to be equally dominant. So if I was going to kind of make my spectrum here, so IA and IB are equally dominant, but both of them are more dominant than the recessive little I. Okay, so let's write in our blood types, and this is something you can fill in your table. So for type A blood, individuals can be IA, and be expressing A surface antigens. They can also be carrying the recessive allele, so they could be heterozygous and expressing it that way. B, it could be homozygous, okay, or heterozygous carrying the recessive allele. Here's our codominant, IA, IB. Both dominant alleles are expressed, so they express both A and B antigens on the surface of red blood cells. And then you can see none of those little surface protein, those little antigens on the surface are expressed. So you don't see any of them with O, so it would be little I. There is another blood type gene. I'm sorry, I jumped ahead. So if we were going to set up our Punnett square, same thing we've been doing before. Okay, if you have a parent that's A and they happen to be carrying the O allele, or B and carrying the O allele, okay, they can give both alleles, and you can see how like two parents can potentially produce four different blood types in their family, which is pretty cool. But we're going to set up Punnett squares just the same as we have been. Okay, there is one more gene, if you ever heard of Rh factor. So this is like the positive and minus that we add to the blood types. So like you could be AB positive or you could be O negative. So this is another surface antigen that's coded for by a single gene, but this one happens to have complete dominance. So if the Rh antigen is expressed, okay, we would say they're positive. If it's not, we would say that they are negative. And we typically tend to use the letter G to represent this gene. So someone who is positive could be big D, or big D, little d. So someone who is negative would be little d. Okay, so we're going to do a little practice with this. You have a couple of practice problems for blood type in your packet. You also have a couple of additional problems that link together, kind of look at Tay-Sachs and sickle cell anemia, and then I have a really good challenge problem in your packet for you to do. So I would love for you to work on and complete up to number 23 in your packet. So do these blood type problems, go to number 23, and then if you are thinking of taking AP Biology next year, I highly recommend that you do that challenge problem. And I recommend that everyone else try it because it is kind of a fun one to solve. Your homework is to have everything up to page 25 finished in your packet, and we will go through these next time. I do want to remind you that there is an answer key online, so if you would like to check any of these as you're going through, you can definitely check them, and we will go through any that you have specific questions about next time. But I do appreciate you working on this in my absence, and I will come visit me if you have any questions or need some help. So have a wonderful day.